Have you ever found yourself maybe in a doctor's office or just, scrolling through a health article online and you bump into terms like pH imbalance
Hmm.
or maybe those acid-producing fat byproducts, ketone bodies,
Right.
and you just thought, what are they actually talking about? Like fundamentally, or maybe you're just really curious about what's going on deep inside your own body, way beyond what you can see, wondering how something as simple as, well, water or a tiny little molecule can dictate health or disease.
Yeah,
Well, if that sounds like you, you're definitely in the perfect place for this deep dive.
absolutely.
Right, let's really get into this essential groundwork. We're diving into section 2 of Mark's Basic Medical Biochemistry, the focus, chemical and biologic foundations of biochemistry.
Crucial section.
Utterly foundational, isn't it? It basically lays down the entire molecular blueprint. If you want to understand everything else in biochemistry and, frankly, a whole lot of medicine, you need this.
You really do. It's the bedrock.
So, our mission today is to pull out the most important nuggets, the key takeaways from the chapters here. Chapter 4 on water, acids, bases, and buffers. Chapter 5, looking at the structures of the major compounds of the body. And Chapter 6, which covers amino acids in proteins.
Hmm.
Think of this as maybe your shortcut, a way to truly grasp the molecular basis of life and disease. Getting this fundamental understanding will honestly make future topics just click, exponentially clearer, more coherent.
And what's really fascinating about this particular section is how these three chapters, even though they seem to focus on different things, water, then big molecules, then amino acids, they're so deeply critically interconnected.
Right, not just separate facts.
Exactly. We're not just memorizing isolated bits of information here. We're actively trying to build a comprehensive mental framework, which, naturally leads us to ask, how do these seemingly simple concepts, like, say, water's unique properties or maybe the electrical charge on a single aminic acid, how do they ultimately orchestrate these incredibly complex physiological functions?
Yeah, the big picture stuff.
And crucially, how do they manifest as the clinical conditions we actually see in patients? We'll be trying to peel back those layers, that why it matters, throughout our entire deep dive today.
Okay.
Ultimately, you'll see that understanding the basic chemistry of these foundational molecules, their structures, how they interact, the delicate environment they operate in, it's absolutely essential. It's the key to making profound sense of both health and disease.
Okay, and this is where the complexity, but also the elegance, I think, really shines. We often take water completely for granted. It's just water, isn't it?
Deep simple.
But biochemically, honestly, it's nothing short of a superstar. It performs this, dizzying array of essential tasks. Your body, on average, what is it? About 60% water by weight.
Roughly, yes. Give or take.
And it's not just sitting there passively. It acts as the primary indispensable solvent. It dissolves countless essential substances, things like vital potassium ions, glucose for energy, ATP.
The energy currency.
Exactly, the universal energy currency of the cell. And a huge array of proteins, too. This amazing dissolving power is precisely what facilitates transport all around the body. It moves everything, nutrients, waste products.
Effortlessly.
Plus, it's absolutely critical for regulating our internal body temperature. Keeps us stable.
So what makes water such an incredible, almost, magical solvent?
Yeah.
It really boils down to its unique structure. Water is what we call a dipolar molecule.
Bipolar, okay.
So imagine an oxygen atom bonded to two hydrogen atoms. The oxygen atom is a bit greedy for electrons. It pulls them closer,
Okay.
giving it a slight partial negative charge. And that leaves the hydrogen atoms with slight partial positive charges.
Though it's uneven.
Exactly. An even charge distribution. And this inherent polarity allows individual water molecules to form these weak but incredibly numerous non-covalent bonds called hydrogen bonds with other polar molecules.
Right, hydrogen bonds. I remember those.
They're like tiny molecular magnets, constantly forming, breaking, reforming. It's this continuous dance that enables water to dissolve so many different organic and inorganic compounds in the body.
Okay, makes sense.
And then when you think about how water keeps our body temperature stable, that's thanks to its high heat capacity and high heat of vaporization. Meaning it simply takes a lot of energy to raise water's temperature or to turn it into vapor like sweat. So it acts like a natural thermostat for our internal environment. Resists temperature changes.
Got it. Buffers the temperature.
Precisely. Now, zooming out a bit, the broader implication here is that water isn't just a uniform sea inside us. It's actually meticulously organized into distinct fluid compartments.
Right, it's not all just sloshing around.
No, definitely not. For an average 70-kilogram adult, the total body of water is around 40 liters.
40 liters, wow.
And this water is cleverly distributed. About 25 liters, so roughly 2/3, is found within the cells. That's our intracellular fluid, or ICF.
Inside the cells, okay.
The remaining 15 liters is extracellular fluid, or ECF, outside the cells. And that's further subdivided.
How so?
Well, about 2/3 of that ECF, roughly 10 liters, is the interstitial fluid. That's the fluid that bathes and surrounds all our cells.
The environment they live in.
Exactly. And the final 5 liters is plasma, the crucial fluid component of our blood, constantly circulating.
Okay, ICF, interstitial fluid, plasma. Got it.
Now, what's truly remarkable and absolutely vital for health is how water moves between these compartments.
It's not static.
Not at all.
Right.
This movement is largely governed by osmosis and the resulting osmotic pressure. Think of a cell membrane like a semi-permeable fence.
Letting some things through, but not others.
Right. And water will always naturally move from an area where there's a lower concentration of dissolved stuff solutes, so lower osmolality, to an area with a higher solute concentration. It's always trying to achieve equilibrium, balance things out.
Okay.
And this principle has profound clinical significance. Consider, for example, hyperglycemia, high blood glucose.
Like in diabetes.
Exactly. The increased solute concentration, the sugar, in the extracellular fluid acts like a powerful magnet. It relentlessly pulls water out of the cells.
Dehydrating the cells.
Precisely. Cellular dehydration. And that can profoundly impair bodily functions, leading to many serious medical complications. So understanding osmosis isn't just academic, it's critical for understanding disease.
Okay, so water sets the stage, moves between compartments. What does this mean for the body's internal stability? You mentioned pH earlier.
Right, it means pH is absolutely everything. Our bodies are constantly churning out metabolic acids just as a byproduct of staying alive. And without extraordinarily tight regulation of pH, our internal environment would quickly spiral into chaos. So let's clarify some terms. Acids are substances that release hydrogen ions, H+ GAN. They donate H+ down.
Okay, acid equals H+ donor.
And bases, on the other hand, are substances that accept those H+ ions. Or sometimes they release hydroxide ions, OH.
Base equals H+ acceptor. Got it.
Now, the concentration of these hydrogen ions is expressed using the pH scale, which is simply the negative logarithm of the H+ concentration.
So lower pH means more H+1.
Exactly, more acidic. Pure water, perfectly neutral, has a pH of 7.0. But in our bodies, the normal physiological pH range for arterial blood is incredibly narrow, just between 7.36 and 7.44.
Wow, that's a tiny window.
It really is. Even slight deviations outside this tiny range can be life-threatening. A drop too low, that's acidosis. A rise too high, that's alkalosis.
Acidosis, alkalosis, okay.
Acids also vary in their strength. Strong acids, like hydrochloric acid in our stomach or sulfuric acid, they dissociate almost completely in water. They dump nearly all their H+ ions.
All in.
Right. But most acids relevant in the body, things like lactic acid from intense exercise, or those ketone bodies we mentioned, acetoacetic acid and hydroxybutyric acid, or even carbonic acid, which is central to breathing. These are weak acids.
Weak acids.
Meaning they only partially dissociate. They exist in this dynamic, reversible equilibrium with their conjugate base. They hold on to some of their H+ fleta.
Okay.
We quantify the strength of a weak acid using its pi value. That's the negative logarithm of its acid dissociation constant, conjugate. The key point here, a lower pKi indicates a stronger weak acid. It's more willing to give up its H+ len.
Lower pKi, stronger acid. Okay.
For example, most metabolic carboxylic acids have pKi values somewhere between 2 and 5, which means that at our body's physiological pH of 7.4, a significant chunk of them are dissociated. They have released their H+ land, and they're contributing to that overall acid-base balance.
Right. Constantly adding acid to the system.
Constantly. Which brings us to how the body handles it. What's truly ingenious is how the body has evolved these sophisticated, almost miraculous systems to maintain this crucial pH balance, despite that constant metabolic acid production. These are our buffers.
The shock absorbers.
Exactly. Think of a buffer as a molecular shock absorber. It's a clever mixture of a weak acid and its conjugate base. This dynamic duo allows the solution to gracefully resist changes in pH when small amounts of H+ or OH ions are inevitably added.
How does that work?
Well, the weak acid component steps in to neutralize any added base, the OH, and the conjugate base readily mops up any added acid, the H+ ion.
So it works both ways.
Precisely. The mathematical blueprint for how a buffer's pH relates to its pKa and the ratio of its conjugate base to weak acid concentrations. That's described by the Henderson-Hasselbalch equation.
Ah, The famous equation.
Indeed. And what it tells us practically is that a buffer works best. It's most effective when the pH of the solution is very close to its pKa, typically within about 1 pH unit.
Why then?
Because at that point, you have roughly equal and ample amounts of both the weak acid and its conjugate base ready to react, ready to neutralize incoming acid or base.
Okay, makes sense. You need both forms available.
Exactly. Now, the human body relies on several major buffer systems working together to maintain this life-sustaining pH homeostasis. The bicarbonate buffer system is arguably the most vital, especially in the extracellular fluid.
Bicarbonate, that involves CO2, right?
Absolutely. It's a constantly reversible reaction. Carbon dioxide, CO2, from our metabolism combines with water to form carbonic acid, H2CO3,
Okay.
which then rapidly dissociates into a hydrogen ion, H plus I, and a bicarbonate ion, HCO3. There's an incredibly fast enzyme called carbonic anhydrase that speeds up that initial formation of carbonic acid.
Speeds it right up.
Dramatically. What's brilliantly adaptive about this system is its open-ended nature. Our lungs continually regulate the amount of CO2. We can breathe faster or slower.
Breathe out the acid, essentially.
Effectively, yes.
Yeah.
By exhaling CO2, we reduce carbonic acid and thus remove H plus from the body.
Yeah.
Simultaneously, our kidneys precisely regulate the concentration of bicarbonate. They can excrete excess H plus and regenerate precious bicarbonate.
Lungs and kidneys working together.
It's this exquisite dual control that makes the bicarbonate system remarkably robust and adaptable.
Wow. Okay, what other systems are there?
Well, beyond bicarbonate, we also have the phosphate buffer system. This plays a particularly significant role inside our cells, intracellularly, because phosphate concentrations are much higher there.
Okay, phosphate inside cells?
And then there's the hemoglobin buffer system, which is absolutely crucial within our red blood cells.
Hemoglobin, the oxygen carrier, it buffers too.
It does, particularly deoxyhemoglobin, the form without oxygen bound. It can efficiently bind H plus ions, acting as a powerful buffer right there in the red blood cells, mopping up acid produced in tissues.
Fascinating.
And these buffer systems are constantly, tirelessly working because normal metabolism, just keeping us alive, generates significant amounts of acids every single day. Lactic acid, ketone bodies, various inorganic acids. Without these intricate, interconnected buffer systems, our bodies would be rapidly overwhelmed by these metabolic acid assaults.
Okay, this is where it gets really practical, I think. Let's explore some real-world clinical examples that really illustrate the importance of all this.
Good idea.
First, let's consider diabetic ketoacidosis, or DKA. This is a severe complication, often seen in people with type 1 diabetes. Let's imagine a patient, Diane A., 26 years old.
Okay.
In DKA, there's an absolute deficiency of insulin. Without insulin, cells can't properly take up glucose for energy.
They're starving for sugar, even though it's high in the blood.
Exactly. So the body, desperately seeking fuel, shifts to metabolizing fatty acids in the liver. This produces large quantities of ketone bodies, primarily acetoacetic acid and hydroxybutyric acid.
And as we said, those are weak acids.
Right. And when they dissociate, they flood the bloodstream with H plus ions, drastically lowering both blood and cellular pH. This leads to severe, potentially life-threatening acidosis.
So what symptoms would Diane have?
She'd likely experience classic symptoms: vomiting, profound dehydration, low blood pressure, and rapid deep respirations, sometimes with a distinctive fruity odor from acetone, which is a breakdown product of acetoacetate.
And the lab results.
Alarming. Blood pH might be 7.08, critically low. Blood glucose could be soaring, maybe 648 milli-GDL, and high levels of ketone bodies in both blood and urine.
A clear picture of DKA.
Now, in response to this dangerous pH plummet, her body initiates powerful compensatory mechanisms. You'd likely see something called Kussmaul breathing.
Deep, rapid breathing.
Exactly. It's a type of hyperventilation that dramatically increases CO2 exhalation. By blowing off CO2, the body effectively reduces the amount of carbonic acid in the blood, desperately trying to raise the pH back up.
The bicarbonate system trying to compensate.
Precisely. The kidneys also rally, working overtime to excrete excess H plus and ammonium ions. Treatment for Diane would involve giving insulin immediately to stop ketone body production and allow cells to use glucose again, plus lots of intravenous saline to correct the severe dehydration and the osmotic diuresis caused by the high glucose.
So DKA isn't just about high sugar. It's a metabolic acid base emergency.
Absolutely. It's a stark reminder that when the body can't access its main fuel, glucose, its backup system, fat burning, can unleash a flood of acids that quickly overwhelm its defenses.
Awesome.
Okay, let's look at another scenario. Dennis V, a 3-year-old who tragically ingested a lot of aspirin, acetylsalicylic acid.
Oh, dear, dear.
Aspirin gets metabolized in the body to salicylic acid, which is a strong acid, so that directly causes a metabolic acidosis.
Okay, straight acid ingestion, essentially.
But aspirin also has this fascinating kind of dual effect. It stimulates the respiratory control center in the brain,
Leading to?
hyperventilation. It makes Dennis breathe too fast, blowing off excessive amounts of CO2.
So that would cause respiratory alkalosis.
Yeah.
Less CO2 means less carbonic acid, higher pH.
Exactly. So here you have this incredibly complex mixed acid-based disorder, both metabolic acidosis from the salicylic acid and respiratory alkalosis from the hyperventilation happening at the same time. Makes diagnosis and treatment really tricky.
A real challenge.
And one more quick example. Percy V, maybe someone experiencing hyperventilation during a panic attack.
Right.
In this case, the excessive exhalation of CO2 leads to a rapid decrease in carbonic acid in the blood. This shifts the bicarbonate buffer equilibrium, causing a rapid increase in pH. The result? Respiratory alkalosis.
Just from breathing too fast.
Just from breathing too fast. So these clinical vignettes, I think, they vividly illustrate how intricately the body's pH is regulated, how even subtle chemical changes or external factors can have profound effects, and how the body heroically uses compensatory mechanisms, water shifts, buffer systems, changes in breathing to try and maintain that fragile balance.
Which naturally brings up the question, what about other dissolved particles in these fluid compartments? And how does the body manage the long-term disposal of excess acid?
Good point. It's not just H plus S.
No. Beyond pH, the body meticulously controls the distribution of electrolytes. These are those crucial ions like sodium, Na+, potassium, K+, chloride, Cl, bicarbonate, HCO3, and phosphates. And it's fascinating how their concentrations differ so much between compartments. Sodium and chloride, for instance, are mostly extracellular. Potassium and phosphates are largely concentrated inside our cells.
Right. Different environments inside and out.
Absolutely. And these electrolytes are indispensable for everything from nerve impulses and muscle contractions to, of course, that intricate dance of water balance we discussed earlier via osmosis.
So they're crucial players. And long-term acid removal.
That's primarily the job of our kidneys. These incredible filtration and reabsorption machines play a fundamental long-term role in regulating the overall H+ content.
How do they do that?
They do it cleverly by excreting ammonium ions, NH4+, and phosphate ions into the urine. This isn't just simple waste disposal. It's a critical, sustained mechanism for maintaining pH homeostasis over the long haul, beautifully complementing the rapid-fire action of the buffer systems.
So buffers for the short term, kidneys for the long term.
That's a good way to think about it. And to bring this to a practical level, consider scenarios of significant fluid loss. Think about how persistent vomiting, losing hydrochloric acid from the stomach, can actually lead to alkalosis, a higher pH.
Right, losing acid makes you more basic.
Exactly. And conversely, how severe diarrhea, where you lose a lot of bicarbonate from the intestines, can lead to acidosis, a lower pH. It's all about disturbing those delicate balances of H+ and bicarbonate, and the body's ceaseless attempts to restore equilibrium.
Right, okay, let's pull back the curtain now on this next huge topic. The sheer variety and immense importance of the organic molecules that literally form the fabric of our bodies.
The building blocks.
Exactly, it's like an alphabet forming words.
Huh.
These compounds, carbohydrates, lipids, nitrogen-containing compounds, they combine to form the incredibly complex language of life. And a core universal principle here, I think, is that the intricate structure of a molecule absolutely dictates its function.
Right.
A tiny tweak in shape or composition can totally change what it does.
Absolutely critical concept.
Yep.
Structure equals function. And what's genuinely captivating about these compounds is that their reactivity, their distinct properties, are largely determined by their specific functional groups.
Functional groups, remind us.
These are specific arrangements of atoms within a larger molecule that are responsible for its characteristic chemical reactions. They act almost like a molecular signature, telling you how that part of the molecule might behave.
Okay, give us some examples.
Sure. If you imagine a chart of major functional groups, among the carbon-oxygen groups, you've got the alcohol or hydroxyl group, just AUH. That makes things soluble reactive. Then you have aldehydes, DSCHO, and ketones, CO even a chain. You find those often in sugars. Carboxylic acids have that base OH group, which, as we just discussed, acts as a weak acid.
Right.
And then more complex ones like esters, ethers, acid anhydrides. Sulfur carbon-sulfur groups, you find out the sulfhydryl group, SH, super important to proteins, and the disulfide bond, a strong covalent link.
Okay.
For carbon-nitrogen groups, we see the amino group, base NH2, the amide group, decoNH2, and quaternary imines. These functional groups aren't just decorative. They are the actual sites where biochemical reactions happen.
Like connecting points.
Exactly. For instance, an alcohol and a carboxylic acid can easily react to form an ester bond that's common to making lipids.
Yeah.
We also constantly see oxidation and reduction reactions in our bodies.
Oxidation is losing electrons.
Basically, yes. Often involves gaining oxygen or losing hydrogen. Reduction is the opposite, gaining electrons or losing oxygen or gaining hydrogen. Understanding these specific groups helps us profoundly predict how molecules will behave and interact in that complex cellular soup.
Okay, that makes sense. Functional groups determine reactivity. So let's apply that. Let's talk carbohydrates, our primary energy source.
Our go-to fuel, yes. These are essentially sugars. And we classify them in a couple of key ways. First, by their carbonyl group, that CO double bond. If it's an aldehyde group, DHCHO, at the end of the chain, it's an aldose.
Aldose, like glucose.
Exactly. Glucose is the classic aldose. If the CO is within the carbon chain, it's a ketose.
Ketose, like fructose.
Perfect example. Fructose is ketose. They're also classified by the number of carbon atoms. Trioses, 3 carbons. Pentoses, 5 carbons, like in RNA and DNA. And hexoses, 6 carbons, like glucose and fructose.
Tripenthex.
Okay, things get a little more intricate and really fascinating with stereoisomers.
Isomers meaning same formula, different arrangement.
Precisely, same chemical formula, but a different 3D arrangement of atoms. A special type is an enantiomer. These are non-superimposable mirror images of each other, like your left and right hands. And in human biology, it's a remarkable fact that we almost exclusively use D sugars, not their L-form mirror images. The enzymes are specific for the D form.
Wow. D sugars.
Now, most simple sugars, monosaccharides, don't actually hang out as linear chains in solution. They cleverly form stable ring structures.
Rings, right. More stable.
Generally, yes. Specifically, 6-membered pyranose rings or 5-membered furanose rings. And within these rings, that carbon atom that used to have the carbonyl group, the anomeric carbon, can exist in 2 distinct forms, alpha and beta anomers. They can flip between these forms in solution through a process called mutarotation.
Alpha and beta, that matters.
Hugely. That subtle difference is absolutely key to how these sugars link together and how other molecules recognize them.
Okay, so these individual monosaccharides link up.
Yes, they're the building blocks. They link together via glycosidic bonds to create larger carbohydrates. 2 linked together make a disaccharide, like lactose, milk sugar. Many linked together form vast polysaccharides.
Like starch.
Exactly. Starch, which plants use for energy storage, and glycogen, which is how animals, including us, store glucose, mainly in the liver and muscles. You can imagine these forming complex branched chains.
Okay. Starch and glycogen.
Furthermore, sugars aren't always just plain sugars. They can be substituted with other chemical groups. They might have phosphate groups attached, like glucose 6-phosphate, which traps glucose inside cells, or amino groups, forming things like N-acetylated sugars, important in connective tissue, or even sulfate groups. And then there's deoxyribose, a sugar in DNA, where one hydroxyl group on the ribose ring is simply replaced by a hydrogen. That small change makes DNA more stable than RNA.
Wow, tiny changes, big effects.
Absolutely. Clinically, as we saw, altered carbohydrate metabolism is central to DKA. And the fact that aldoses are reducing sugars they can react in certain tests allows them to be detected in urine, which is still important for monitoring glucose in some contexts. The real marvel isn't just the variety, but how these seemingly simple variations in structure, aldose versus ketose, DVSL, alpha versus beta, completely alter recognition and function, determining if it's fuel, structure, or something else entirely.
Fascinating. Okay, what about the next group? Lipids, fats, and oils.
Right. If we connect this to the bigger picture, lipids are incredibly diverse and utterly crucial for everything from cell structure and signaling to very efficient energy storage. Unlike carbs, lipids are largely hydrophobic.
Water-fearing, they don't mix.
Exactly. Oil and water don't mix. The simplest lipids are fatty acids. They have a pretty straightforward structure. A long, straight aliphatic carbon chain with a methyl group, CH3 at one end, and a carboxyl group, -COH at the other.
The acidic part.
Yes. Fatty acids come in 2 main types. Saturated, meaning they have no double bonds in their carbon chain. They're straight, flexible chains.
Saturated, no double bonds.
Okay, and unsaturated, meaning they possess 1 or more double bonds in the chain.
Unsaturated have double bonds.
Now, the precise configuration, the geometry of these double bonds is remarkably important. Most naturally occurring unsaturated fatty acids have cis double bonds.
Cis.
Cis double bonds introduce a distinct kink or bend in the fatty acid chain. Imagine a sharp turn. This means they can't pack together as tightly.
Okay, cis means kinked, less packed.
Right. In contrast, trans double bonds allow the carbon chain to remain relatively straighter, more like a saturated fatty acid. This allows for tighter packing.
Trans means straighter, packs tighter, like the infamous trans fats.
Exactly the ones. You find trans fats in partially hydrogenated oils. They're particularly detrimental to health, significantly increasing the risk of heart disease, partly because their straight, tightly packing nature can disrupt cell membrane fluidity and lipid metabolism.
Okay, avoid trans fats. What about omega fatty acids?
Good question. We also have omega fatty acids like omega-3 and omega-6. The name tells you where the first double bond is, counting from the methyl omega end. These are essential nutrients we have to get them from our diet.
Why are they essential?
They serve as crucial precursors for vital hormone-like compounds called eicosanoids, things like prostaglandins and leukotrienes, which are involved in inflammation, blood clotting, immune responses. Really important signaling molecules.
Got it. Omega-3s and 6s are starting materials.
Precisely. Now, beyond simple fatty acids, we have triacylglycerols, more commonly known as triglycerides.
Triglycerides, hear that a lot in blood tests.
You do. These are formed when three fatty acids are chemically bonded, esterified, to a glycerol backbone. Their primary role is incredibly efficient long-term energy storage. They pack a huge amount of energy into a small, compact, water-free space, much more energy-dense than glycogen.
Our main energy reserve.
Yes. Then there are phosphoglycerols, or phospholipids. These are similar to triglycerides, but instead of three fatty acids, they have a glycerol backbone, two fatty acids, and a phosphate group. That phosphate is often linked to another polar head group, like choline and phosphatidylcholine, which is a major membrane component.
So 2 fatty acid tails and a phosphate head.
Exactly. And this structure makes phospholipids famously amphipathic.
Amphipathic, both.
Means they have both parts, a hydrophilic, water-loving phosphate head group and hydrophobic, water-fearing fatty acid tails. This dual nature is precisely what allows them to spontaneously assemble in water into the crucial bilayers that form the structural foundation of all cell membranes, creating that vital barrier.
The cell membrane bilayer, driven by phospholipids.
Wow, that's fundamental. We also encounter sphingolipids. These are built on a different backbone molecule called sphingosine, which itself comes from the amino acid serine plus a fatty acid. This class includes important molecules like ceramides, cerebrosides, and gangliosides.
Where are they important?
They are also important components of cell membranes, particularly enriched in the nerve myelin sheath, providing insulation for nerve impulses. Defects in sphingolipid breakdown cause serious diseases.
Okay. And finally, steroids.
Finally, steroids. These are characterized by a distinctive, rigid, four-ring steroid nucleus structure. Cholesterol is the most widely recognized steroid.
Cholesterol gets a bad rap.
It often does, but it's far from just a bad guy. Cholesterol is a vital component of animal cell membranes, influencing their fluidity and stability. It prevents them from being too fluid or too rigid. And crucially, it serves as the precursor, the starting material for all other steroid hormones like estrogen, testosterone, cortisol, and also for bile salts, which are essential for digesting and absorbing fats.
So essential precursor and membrane component.
Absolutely. This incredible diversity in lipid structures from simple fatty acids to complex steroids just highlights their wide-ranging and indispensable functions across pretty much every aspect of biology.
Okay, that's a lot for lipids. What about the nitrogen-containing compounds? That sounds important for genetics and things.
Extremely important. So we're moving into nitrogen-containing compounds. We've briefly mentioned amino acids already, the building blocks of proteins. We'll dive much deeper into them next. Just to recap, they all contain both an amino group NH2 and a carboxyl group COH, plus that unique side chain, the R group.
Amino acids, got it. What else?
Beyond amino acids, we had the purines and pyrimidines. These are fascinating heterocyclic nitrogen-containing ring structures, absolutely fundamental to all life.
Purines and pyrimidines, the bases in DNA and RNA.
Exactly. Examples of purines, which are the larger double-ring structures, are adenine A and guanine G.
A and G.
Pyrimidines are smaller, single-ring structures. These include cytosine, C, thymine, T, found in DNA, and uracil, U, found in RNA.
C, T, and U. Okay.
Now, when a purine or pyrimidine base is attached to a sugar, either ribose for RNA or deoxyribose for DNA, and a phosphate group, or maybe more than one phosphate, you get a nucleotide.
Ah, nucleotide equals base plus sugar plus phosphate.
Precisely. Think of ATP, adenosine triphosphate, as the prime example. It's an adenine base, a ribose sugar, and 3 phosphate groups linked together. Nucleotides are the ultimate building blocks of DNA and RNA, carrying the genetic blueprint. And ATP is also the main energy currency.
Building blocks and energy.
Yeah.
Okay, you mentioned something about tautomers.
Yes. Cautomerism is a clever molecular trick we see in these nitrogen-containing rings like uracil or guanine or even uric acid. Think of tautomers as molecular shapeshifters.
Shapeshifters.
It's a rapid equilibrium where the molecule can switch between 2 slightly different forms, usually by a hydrogen atom jumping places and rearranging a double bond. This subtle dance can drastically change a molecule's properties, like its ability to form hydrogen bonds or even its solubility.
Solubility,
Yeah.
like with uric acid and gout.
Exactly. The less soluble tautomer of uric acid is more prone to crystallizing out in the joints, which is the basis of gout. So tautomerism isn't just a chemical curiosity, it has real biological consequences.
Okay, wow. So these are the main players. But what about when things go wrong chemically? You mentioned free radicals.
Right. Which naturally leads us to ask, what happens when these fundamental chemical foundations go awry, or when highly reactive species get introduced into this delicate cellular environment? This brings us to free radicals.
Reactive oxygen species, things like that.
Exactly. These are compounds characterized by having an unpaired electron in their outer shell. That makes them incredibly unstable and, consequently, highly reactive. Common examples in biology include the superoxide anion, O2, and the hydroxyl radical HO, which is extremely reactive.
What do they do?
Because they're so reactive, they desperately want to grab an electron from somewhere else to become stable. In doing so, they can wreak havoc by indiscriminately damaging essential cell components, DNA, proteins, lipids, and membranes.
So collateral damage. Significant collateral damage. This widespread cellular damage, often called oxidative stress when the damage overwhelms defenses, is strongly associated with the development and progression of many diseases: coronary artery disease, diabetes, arthritis, cancer, neurodegenerative diseases like Alzheimer's.
So a major factor in aging and disease.
It's thought to be, yes. While the body does have its own powerful antioxidant defense systems to neutralize free radicals, an imbalance where radical production outstrips the defenses can lead to significant and detrimental cellular harm.
Okay, free radicals is bad news if uncontrolled. Now let's connect these chemical concepts to some more clinical scenarios. Understanding these molecular properties seems vital for understanding disease.
It really is.
Let's talk about some infamous environmental toxins, things like DDT, dioxins, PCBs.
Right. Persistent organic pollutants.
DDT, dichlorodenafenyltric chloroethane, a chlorinated aromatic hydrocarbon, once widely used as an insecticide. Its chemical structure explains why it's so problematic. First, it's incredibly persistent in the environment. Doesn't break down easily.
Stays around for ages. And second, it's highly lipophilic.
Lipophilic meaning fat-loving.
Exactly. It dissolves readily in fats and oils, not water. This allows it to bioaccumulate. It builds up in the fatty tissues of organisms, getting more concentrated as you move up the food chain.
Litomagnification.
Right, leading to long-term neurotoxic effects and famously causing thin eggshells in birds of prey, which was devastating. Then we have dioxins, specifically polychlorinated dibenzo-p-dioxins. These aren't usually made intentionally. They're incredibly toxic byproducts of industrial processes like incinerating waste, burning fossil fuels, pulp and paper manufacturing.
Unwanted side products.
Highly unwanted and extremely toxic. Their toxicity is compounded by their extraordinarily long half-life, and humans could be 5 to 15 years or even more.
Wow, they really stick around.
They do, and they can cause severe skin lesions like chloracne, developmental defects, cancer, immune suppression. Nasty stuff. Similarly, PCBs, polychlorinated biphenyls, were once widely used in things like electrical transformers and capacitors because they're stable and non-flammable. But like dioxins, they bioaccumulate and can significantly affect the immune, reproductive, nervous, and endocrine systems. The crucial common thread binding these toxins together.
Their lipophilicity.
Exactly. Their profound hydrophobicity and lipophilicity. It allows them to concentrate and persist in our adipose tissue, our body fat, for extended periods, posing severe long-term health risks. Understanding their chemical nature explains their biological danger.
Definitely. Structure dictates behavior, even for toxins.
Okay, let's shift gears to a patient example. Gout, as seen in maybe Lotta T. She experiences gouty arthritis, severe, often excruciating pain, frequently starting in the big toe.
Classic presentation.
This painful inflammatory condition is caused by abnormally high levels of uric acid in the blood. Uric acid, which we saw earlier, is a breakdown product of purine metabolism, those A and G bases.
Right, purine breakdown leads to uric acid.
And uric acid, especially in one of its tautomeric forms, isn't very soluble. When its concentration in the blood gets too high, either because too much is made or not enough is excreted by the kidneys, it starts to precipitate out. It forms insoluble urate crystals.
Like tiny sharp needles.
Exactly. These sharp monosodium urate crystals then deposit in the joints and surrounding tissues, triggering an intense, painful, inflammatory response that's the gout attack.
Ouch.
Understanding the metabolism of purines and the critical solubility properties of uric acid, including those tautomers, is absolutely key here. Treatment often involves drugs like colchicine to reduce the acute inflammation and maybe allopurinol.
Allopurinol inhibits uric acid production, right?
Precisely. It blocks an enzyme called xanthine oxidase, lowering uric acid levels and preventing further crystal formation. So these examples, from environmental toxins concentrating in fat to uric acid crystals forming in joints, powerfully demonstrate how understanding the fundamental chemical properties, lipophilicity, solubility, structure, metabolism, is utterly crucial for both understanding disease origins and guiding effective treatment.
So after laying down all that foundational chemistry water, pH, the basic molecules like carbs and lipids, the bases, what's the grand culmination? Where does it all lead?
Good question. We arrive at proteins, the ultimate multitaskers, wouldn't you say? The molecular machines that perform virtually every single cellular function.
Truly the workhorses of life.
Think about it. They act as transporters, diligently moving stuff like oxygen, hemoglobin, and nutrients across membranes. They are the enzymes that catalyze nearly all biochemical reactions, making life happen at speed.
Without enzymes, reactions would be far too slow.
They function as hormones, carrying signals between cells. They form critical structural components like collagen in skin and bone, keratin in hair, giving tissues shape and strength. They're essential to our immune responses, antibodies or proteins.
The list goes on and on. Muscle contraction, light detection.
It's amazing. And the almost unbelievable thing is that all this incredible diversity and functionality stems from proteins being linear chains of just 20 different amino acids.
Only 20 standard building blocks.
Which then meticulously fold into incredibly complex and precise three-dimensional structures. And the specific order of these amino acids, what we call the primary structure, is meticulously dictated by our genetic code, by DNA.
Which brings us to the amino acids themselves. What's genuinely captivating here is the elegant simplicity of the basic amino acid structure, which somehow underpins that incredible, almost infinite diversity of proteins.
So what's the basic structure?
Let's look at the general structure of an alpha amino acid. Every single one shares a common core. There's a central carbon atom, called the alpha carbon.
Okay, central alpha carbon.
Attached to this central carbon are 4 distinct groups.
Oh.
First, an amino group, -NH2. Second, a carboxyl group, H2OH.
Amino and carboxyl. Got it.
Third, a single hydrogen atom. And fourth, crucially, a unique side chain, often just abbreviated as the R group.
The R group, that's the variable part.
Exactly. It is this R group that truly differentiates each of the 20 standard amino acids, giving each one its own distinct chemical properties, its size, its charge, its polarity, its reactivity, its personality, if you will.
20 different personalities.
Now, an absolutely vital concept to grasp is that of deuterions.
Deuterions. Sounds complex.
It just describes their state at physiological pH, around 7.4 in body. At this pH, the amino group is typically protonated. It picks up an H+ and becomes positively charged, NH3+.
Okay, amino group is positive.
Simultaneously, the carboxyl group is usually deprotonated. It loses its H+ and becomes negatively charged. That carboxyl group is negative. Right. So while the molecule as a whole might be electrically neutral overall, one positive, one negative charge, it precisely possesses both a positive and a negative charge at the same time. That's a deuterion.
Both positive and negative charges on the same molecule?
Exactly. And the pK values confirm this. The alpha carboxyl group typically has a pK around 2, and the alpha amino group has a pK usually between 9 and 10.
Yeah.
Meaning that at our body's neutral pH of 7.4, the carboxyl group, pK at 2, is definitely deprotonated, negative, because 7.4 is way above 2. And the amino group, pKi 910, is definitely protonated positive because 7.4 is well below 910.
So both ends are charged at body pH?
Almost always, yes. And this inherent dynamic charge isn't just a chemical detail. It's precisely why proteins are such incredibly dynamic interactive molecules. These charges are crucial for how proteins fold, how they interact with each other, with water, with other molecules, and ultimately how they perform their specific biological roles.
Okay, the charges are key, and the R groups give them their unique properties.
Absolutely. This is where the complexity truly shines. Those R groups, the side chains, are what give each amino acid its unique personality and dictate how proteins ultimately fold and interact. If you were looking at a chart categorizing them,
How would they be grouped?
they're cleverly grouped by the distinctive properties of their side chains. First, you have the nonpolar aliphatic amino acids. Think glycine, alanine, valine, leucine, isoleucine, and proline.
Nonpolar, aliphatic, hydrophobic.
Largely hydrophobic, yes. They tend to avoid water. Consequently, they're often found tucked away in the interior core of a folded protein, shielded from the watery environment. Proline is a bit special. Its unique cyclic structure actually creates distinctive kinks or bends in the polypeptide chain, influencing the overall protein shape.
Okay, nonpolar inside, proline makes kinks. Next.
Next, there are the aromatic amino acids, phenylalanine, tyrosine, and tryptophan. They have bulky ring structures in their side chains.
Aromatic rings, also hydrophobic.
Generally, yes, they lean towards being hydrophobic and are often involved in stacking interactions within proteins, which helps stabilize the structure. Tyrosine stands out slightly because it has a hydroxyl H group on its aromatic ring, which makes it a bit more polar than phenylalanine. Tryptophan is the bulkiest.
Okay, aromatics often inside too. What about polar ones?
Then we move to the polar uncharged amino acids. This includes serine, threonine, asparagine, glutamine, and cysteine.
Polar, but no charge at pH 7.4.
Exactly. Their side chains contain groups like hydroxyls, or amide-CoN H2, that can readily form hydrogen bonds with water or other polar groups. So they're frequently found on the surface of proteins, happily interacting with the watery cellular environment.
Cysteine is notable here because it contains that reactive sulfhydryl-ACH group.
Cysteine's that H group. Important. Very. Speaking of sulfur, cysteine and methionine are the two sulfur-containing amino acids. Methionine is actually pretty nonpolar and often serves as the starting amino acid in protein synthesis. But cysteine, its sulfhydryl groups, -SH, can react with each other, forming a strong covalent bond called a disulfide bond.
Disulfide bond,
But
like a
not-
molecular stable.
Exactly like that. These bonds are absolutely crucial for stabilizing the intricate three-dimensional structure of many proteins, especially those that exist outside cells, like antibodies or insulin. They lock the folded structure in place.
Okay, disulfide bonds equal structural stability. What about charged amino acids?
Finally, we categorize amino acids by their charge at physiological pH. The acidic, negatively charged amino acids are aspartate and glutamate. Their side chains contain a second carboxyl group, which is deprotonated, negatively charged, at pH 7.4. They often participate in vital ionic bonds or salt bridges within the protein or with other molecules.
Aspartate, glutamate, negative charge, and positive.
The basic, positively charged amino acids are histidine, lysine, and arginine. Their side chains contain nitrogen-containing groups, like amino groups or the guanidinium group in arginine, that are protonated, positively charged at physiological pH. They also readily form ionic bonds.
Lysine, arginine, histidine, able positive charge.
Right. And histidine is particularly unique. Its imidazole ring side chain has a pKa value very close to physiological pH, around 6.0. This means it can easily exist in both protonated positive and unprotonated neutral forms near pH 7.4.
So it can accept or donate protons easily.
Exactly. This makes histidine an excellent buffer within proteins and often critically important in the active sites of enzymes where proton transfer is needed.
Wow, okay. And there's a scale for hydrophobicity?
Yes, the hydrophobicity index. It's a quantitative measure, basically assigns a number to each amino acid based on how likely it is to interact with water versus burying itself away. This tendency, hydrophobic residues burying themselves, hydrophilic residues staying on the surface, is a major driving force behind how a protein folds into its specific functional shape.
The hydrophobic effect driving folding. Makes sense.
Stepping back then, we see the profound interconnectedness. These individual amino acids, with their diverse properties, link together to form the very backbone of a protein. How does that linkage happen?
The peptide bond.
Exactly. This linkage occurs through a crucial process called peptide bond formation. A peptide bond is a strong covalent bond formed between the carboxyl group, COOH, of one amino acid and the amino group, NH2, of the next amino acid. with a simultaneous elimination of a molecule of water.
Like linking beads on a string.
It's a good analogy. It's like building a molecular chain, link by link, where each amino acid is a meticulously crafted link. The resulting chain is what we call a polypeptide.
Polypeptide.
And the precise linear sequence of amino acids in this polypeptide chain, that is what we define as the primary structure of the protein.
Just the order of amino acids, A, G, C, V, L, whatever it is.
Precisely. Sequence is everything. This seemingly straightforward sequence is absolutely fundamental. It's the master blueprint because it dictates everything that follows. How that chain will spontaneously fold up into its specific complex three-dimensional shape, its secondary, tertiary, and maybe quaternary structures, and ultimately what its specific biological function will be.
So sequence determines structure. Structure determines function.
That's the central dogma of protein science. Any minute change in this precise sequence can have profound and sometimes devastating effects.
Okay, so what does this all mean when even a tiny change occurs in that primary structure? You said profound effects.
It means small alterations can indeed have massive cascading consequences. This brings us directly to mutations and polymorphisms. Changes in the DNA sequence, the genetic code, which we term mutations, directly lead to changes in the amino acid sequence of the protein being built.
A typo in the DNA blueprint leads to a wrong amino acid.
Exactly.
Yeah.
And even a single seemingly minor amino acid substitution can profoundly affect how the protein folds, how stable it is, or how it interacts with other molecules, potentially impairing or completely abolishing its function.
Give us the classic example.
The classic, incredibly powerful example is sickle cell anemia. Let's imagine Will S., someone with this condition.
Okay.
In this inherited genetic disorder, a single nucleotide change in the DNA code for the beta-globin chain of hemoglobin, just one letter change,
One letter out of billions.
leads to a single amino acid substitution in the protein. Specifically, at position 6 of the beta-globin chain, a negatively charged glutamate residue is replaced by a nonpolar hydrophobic valine residue.
Glutamate swapped for valine. Seems small.
Seemingly tiny. But this single change in the primary structure triggers a dramatic, catastrophic shift in the protein's behavior, specifically when oxygen levels are low. This altered hemoglobin, called hemoglobin SHTS, when deoxygenated, starts to stick together, polymerizing into long, stiff, insoluble fibers.
Polymers inside the red blood cell.
Exactly. These rigid fibers distort the normally flexible, disc-shaped red blood cells into a characteristic crescent or sickle shape.
Ah, the sickle shape.
These sickle cells are rigid, they can't squeeze through tiny capillaries easily, they get stuck, blocking blood flow, leading to excruciating vaso-occlusive crises, tissue damage due to lack of oxygen, ischemia, severe pain.
Awful.
And these fragile sickled cells are also destroyed much faster than normal red blood cells, leading to chronic anemia, all from one amino acid change.
Incredible. But isn't there some advantage? Malaria?
Ah, yes. Remarkably, carrying just one copy of the sickle cell gene, being heterozygous, having the sickle cell trait, actually confers significant resistance to malaria caused by the parasite Plasmodium falciparum. The parasite has trouble developing inside cells with HPS. So in regions where malaria has historically been prevalent, like parts of Africa, the sickle cell trait offered a survival advantage, which is why the mutation became relatively common in those populations. A fascinating evolutionary trade-off.
Wow. Okay, so mutations can have huge effects. What about other variations, isoforms?
Right. Beyond genetic mutations causing disease, proteins can also vary in more subtle yet significant ways. We have tissue-specific isoforms, also known as isozymes if they're enzymes.
Isoforms, lysozymes, different versions in different tissues.
Exactly. Different tissues can express slightly different forms of the same protein, often encoded by different but related genes. These isozymes perform the same overall function, but they might have slightly different kinetic properties, like how fast they work or what substrate concentration they prefer, or they might be regulated by different mechanisms. This allows function to be exquisitely fine-tuned to the unique metabolic needs or conditions of specific tissues.
Okay, clinical example.
A compelling clinical example is seen with creatine kinase, CK, especially in diagnosing a heart attack, an acute myocardial infarction. Let's think about Ann J coming to the ER with chest pain.
Okay.
When heart muscle is damaged, proteins leak out into the blood. Creatine kinase, CK, is an enzyme involved in energy metabolism in muscle. It's composed of two subunits, which can be either M-type for muscle or B-type for brain.
M and B subunits.
These combine to form three main isoforms: CKMM, two M subunits, which is predominant in skeletal muscle; CKBBB, two B subunits, found primarily in brain tissue; and crucially, CKMB1M1B subunit, which is found predominantly in cardiac muscle, heart muscle.
Ah, CKMB is the heart one.
Mostly, yes. So if Ann J has had a heart attack, her damaged heart muscle cells release CKMB into her bloodstream. Measuring elevated CKMB levels in her blood is therefore a highly useful diagnostic marker for heart damage, helping clinicians differentiate a heart attack from, say, skeletal muscle injury, which would release mostly CKMM.
Clever, using isoforms for diagnosis.
Very clever. Proteins also exhibit developmental variations. During different stages of development, like fetal life versus adulthood, specific proteins can have different forms tailored to that stage's needs.
Like fetal hemoglobin.
Exactly. Fetal hemoglobin, HbF, is the primary oxygen carrier during gestation. It actually has a higher affinity for oxygen than adult hemoglobin, HbA. This is absolutely crucial because it allows the fetus to efficiently extract oxygen from the mother's blood across the placenta. After birth, HbF production switches off and HbA takes over.
Higher oxygen affinity for the fetus. Makes sense.
And finally, there are species variations. The primary structure of a protein, like insulin, often varies slightly between different species. Beef insulin, pork insulin, and human insulin have small differences in their amino acid sequences.
Does that matter for treatment?
It used to. Before genetic engineering, diabetics were treated with insulin purified from cows or pigs. But because of those small sequence differences, some patients developed allergic reactions or immune responses. This is precisely why recombinant human insulin, like Humulin or Lispro, was such a breakthrough. It's produced using biotechnology to have the exact human amino acid sequence, minimizing immune issues, like Diane with DKA would receive.
Okay, so sequence matters for function, disease, diagnosis, development, even across species.
Sequence is paramount. But what's genuinely captivating here is that the story of a protein doesn't actually end once its amino acid sequence is synthesized by the ribosome.
There's more.
Oh yes. Proteins frequently undergo further, precise chemical changes after they are initially built. This is called post-translational modification, or PTM.
Post-translation, after the genetic message is translated into protein.
Exactly. These modifications are absolutely essential for a protein's proper function, its correct localization within the cell, where it needs to go, and how its activity is meticulously regulated and fine-tuned. If you were looking at a diagram illustrating PTMs, you'd be struck by their incredible diversity and specificity.
What kind of modifications are we talking about?
One of the most crucial widespread PTMs is phosphorylation. This is where a phosphate group, PO43, is chemically added, typically to the hydroxyl group of serine, threonine, or tyrosine residues.
Adding a phosphate, what does that do?
It often acts like a molecular on-off switch, or maybe a dimmer switch, for enzyme activity or protein interactions. Adding that bulky, negatively charged phosphate group can dramatically change the protein's shape or its ability to bind other molecules. It's a fundamental mechanism for cellular signaling and control, allowing cells to rapidly respond to signals.
Phosphorylation regulation switch. Okay, what else?
Another profoundly important modification is glycosylation. This is the precise addition of carbohydrate groups, sugars, often complex branch chains, to specific amino acids like serine, threonine, olein glycosylation, or asparagine, N-linked glycosylation.
Adding sugars. Why?
This is vital for many proteins, especially those on the cell surface or secreted from cells. It plays key roles in protein folding instability, protecting proteins from degradation, and crucially in cell-to-cell recognition and signaling. Think blood types. Those ABO antigens are determined by specific sugars attached to proteins and lipids on red blood cells.
Glycosylation recognition folding. Got it.
We also encounter acylation and prenylation. These involve the covalent addition of lipid groups like fatty acids, palmitoylation, myristoylation, or isoprenoid groups, prenylation, to proteins.
Adding fats.
Yes. These lipid anchors often serve to tether proteins to specific cell membranes, localizing them where they need to function, like signaling proteins that need to be near the plasma membrane.
Lipid anchors for membrane localization. Okay.
Then there's ADP ribosylation, the addition of an ADP ribose group. This modification is often nefariously carried out by bacterial toxins like cholera toxin or diphtheria toxin. They modify key human proteins involved in signaling, disrupting vital cellular pathways and causing disease symptoms.
Bacterial toxins hijacking PTMs.
That's nasty.
Very nasty. Other key modifications include carboxylation, adding a carboxyl group, COH. This is notably important for certain glutamate residues in blood clotting proteins. That extra carboxyl group allows them to bind calcium ions, Ca2+, which is a critical step in the blood coagulation cascade.
Carboxylation for blood clotting. And hydroxylation, the addition of a hydroxyl group, OH. This is crucial for specific proline and lysine residues in collagen, the main structural protein in connective tissue. Those hydroxyl groups are needed for collagen's cross-linking and incredible tensile strength. Lack of hydroxylation, often due to vitamin C deficiency, scurvy, leads to weak collagen.
Hydroxylation for collagen strength.
There's even a fascinating 21st standard amino acid, selenocysteine. It contains selenium and is incorporated during protein synthesis at specific UGA codons, which usually means stop. It's found in some important enzymes, often those involved in antioxidant defense, like glutathione peroxidase.
Wow, even a 21st amino acid via PTMs. Well, incorporated during synthesis.
Sort of co-translational modification, yes. The key takeaway is that these post-translational modifications are not just random chemical additions. They are absolutely critical for vastly expanding the functional repertoire of the 20 basic amino acids, for fine-tuning protein activity, directing proteins to the right place, and allowing proteins to precisely respond to the complex and ever-changing array of cellular signals they encounter. It's another layer, a crucial layer, of exquisite biological control.
Amazing level of detail and regulation. Okay, let's bring this back to a powerful real-world clinical scenario again. Cystinuria, as observed perhaps in David K., he developed a renal stone, a kidney stone, because of this genetic disorder.
Right, cystinuria.
It's a condition that profoundly affects the kidney tubule cell's ability to properly reabsorb certain amino acids from the fluid that's on its way to becoming urine. Specifically, it affects the transport of cysteine and also 3 basic amino acids: arginine, lysine, and ornithine.
A specific transporter defect.
Exactly. As a result, these amino acids are present in dangerously high concentrations in the urine. Now, arginine, lysine, and ornithine are relatively soluble, but cysteine, remember cysteine?
The one with the NSH group.
Yes. Cysteine is actually formed when 2 cysteine molecules link together via a disulfide bond, SS. And cysteine is relatively insoluble, especially in acidic urine.
Ah, solubility problem.
Big time. When its concentration in the urine reaches a critical point, it precipitates out of solution, forming insoluble hexagonal cysteine crystals. These crystals then aggregate, grow, and form painful kidney stones, or calculi.
Ouch. Caused by insoluble amino acid dimer.
Precisely. These stones can cause severe pain as they pass, block the flow of urine, lead to infections, and potentially cause significant kidney damage over time. Treatment typically involves dramatically increasing fluid intake to try and dilute the urine and keep the cysteine dissolved, sometimes adjusting urine pH, and maybe medications to enhance cysteine solubility. This case vividly highlights how a seemingly specific defect in the transport of even relatively simple molecules, like amino acids and their dimers, can have profound and painful clinical consequences due to basic chemical properties like solubility.
It really connects the basic chemistry to the clinical reality.
Absolutely. Now, just briefly, for students and researchers listening, tackling all this complexity, understanding proteins relies heavily on data, right?
Immensely so. In today's incredibly advanced scientific landscape, truly understanding proteins goes hand in hand with accessing and utilizing vast amounts of publicly available data. That's where protein databases become indispensable.
Like PubMed.
PubMed is crucial for the scientific literature, finding research papers. But for protein-specific data, think of resources like GenBank from NCBI for nucleic acid sequences, which code for proteins. Then there's the RCSB Protein Databank, the PDB.
PDB for structures.
Exactly. It's the worldwide archive for experimentally determined three-dimensional structures of proteins and nucleic acids, incredibly valuable for understanding function. And EMBL, the European Molecular Biology Laboratory, also houses extensive and diverse databases for sequences, structures, and more.
So these databases are essential tools.
Absolutely essential. They allow researchers and clinicians to quickly and efficiently access detailed information on protein sequences, genetic variations, 3D structures, functional annotations, and crucial associations with various diseases. They are indispensable tools for facilitating deeper understanding, accelerating cutting-edge research, and ultimately speeding up the development of new diagnostic tools and innovative drug therapies that can genuinely improve human health. Data sharing is key. #tag connecting the deep dive together.
Okay. So we've covered a lot of ground. Water, pH, buffers, the chemical alphabet of carbs, lipids, bases, and then the amino acids building proteins with all their modifications. How does it all tie together?
Yeah, that's the critical question, isn't it? How do all these foundational concepts intricately interlink? We've really embarked on a journey today. We started with water, the universal solvent, establishing that essential aqueous environment where all biochemical reactions occur. Sword, we saw how its unique properties are intricately influenced by pH, and how those vital buffer systems, especially bicarbonate, tirelessly work to maintain that delicate, life-sustaining balance against constant acid production.
Buffers holding the line.
Then we explored the chemical alphabet, carbohydrates, lipids, and nitrogen-containing compounds. We discovered how their unique structures and specific functional groups dictate their incredibly diverse roles, from providing immediate energy and structural support in carbohydrates,
To forming crucial cell membranes and storing vast amounts of energy in lipids.
and critically, carrying our genetic information in those nitrogen-containing purine and pyrimidine bases. We also touched upon the inherent vulnerabilities, like the indiscriminate damage caused by free radicals or the insidious accumulation of lipophilic environmental toxins, underscoring why understanding these foundational chemical properties is so vital.
Right. Structure dictates function and vulnerability.
And finally, we focused on amino acids. We saw how the precise linear sequence, the primary structure determined by our genes, forms the basis of proteins, and how this sequence, along with that amazing array of post-translational modifications they undergo, creates the diverse army of proteins that perform the vast majority of functions in our bodies.
Everything from transport and catalysis to structure and defense.
Exactly. We learned how even a single amino acid change, so powerfully illustrated in sickle cell anemia, can profoundly alter protein function and cause disease, or how specific isoforms, like creatine kinase CKMB, released during a myocardial infarction, can serve as crucial diagnostic markers because their tissue distribution is unique.
Connecting molecules to medicine?
Precisely. Each layer of this incredible biochemical system meticulously builds upon the last. Water sets the stage, the molecules provide the building blocks and fuel, and proteins do the work. It forms this cohesive and powerful understanding of how life operates at its most fundamental molecular level. And crucially, it illuminates how disruptions at any point within these foundational systems, a faulty enzyme, a mutated structural protein, an overwhelmed buffer system can cascade through, ultimately leading to the complex diseases we encounter in the clinic. It all connects back. #outro.
Well, that was certainly an extensive journey, a real deep dive into the chemical and biologic foundations of biochemistry. We've definitely peeled back many layers today, uncovering some of the molecular elegance that underlies life itself.
It's a fascinating level to explore.
Absolutely. So just to recap some key takeaways. Remember, water isn't just H2O. It's our body's essential solvent. It regulates temperature, distributes substances, all while maintaining incredibly tight control over pH through those vital buffer systems like the bicarbonate buffer.
Can't live without that balance.
We then explored the fundamental structures of carbohydrates, lipids, and nitrogen-containing compounds. We saw how their functional groups and unique shapes give them their diverse, indispensable roles, from energy to membranes to genetic code. And we glimpsed how things like highly reactive free radicals or persistent environmental toxins can really wreak havoc on these delicate chemical foundations.
Understanding the chemistry explains the biology and pathology.
And finally, we saw how amino acids are the incredibly versatile building blocks of proteins. Their specific sequences, dictated by our genes, and then those crucial post-translational modifications determine everything from oxygen transport in conditions like sickle cell anemia to serving as diagnostic markers for heart attacks. It's truly incredible how all these fundamental chemical principles seamlessly come together,
They really do build upon each other in a remarkable way.
creating life as we know it.
Which, raises an important, maybe final question for you, our listeners, to ponder. Considering the intricate balance and the profound interconnectedness we've discussed today, how might a seemingly small change at the molecular level, perhaps just a single amino acid substitution, like in sickle cell, or maybe just a subtle, persistent shift in pH, how might that cascade through these foundational systems to profoundly impact an organism's health, its physiology, or even, thinking bigger, influence its evolutionary trajectory over millennia?
That's a great question to leave people with, the ripple effects of tiny changes.
Exactly.
Keep exploring, keep asking those questions, and definitely keep making those connections between the molecules and the medicine. We really hope this deep dive has given you a clearer, perhaps more informed perspective on the foundational chemistry that truly underlies all biological processes. Thank you so much for joining us on the deep dive.